Hi students, this is video 1 of protein structure and function corresponding to ch4. I've made a series of 4 videos to help you learn some things about proteins. You should have your chapter study questions out and also your textbook to follow along as you go. This first video deals with differences among the 20 common amino acids and how the unique sequence of amino acids is able to impact the way a polypeptide folds. For answers to your first question, you should look at page 122 in your textbook, which has a panel with some interesting examples of some general. You should look at the headings and the general functions. That will give you answers to your questions. For example, as enzymes, they mediate chemical reactions. As structural proteins, they give cells shape, structure, and strength. As motor proteins, they can generate movement either inside the cell or as tissues or even the whole body movement. Other proteins store and transport materials that the cells need. They receive and send signals so the organism can communicate and respond to its environment. And also they regulate the expression of genes according to need and do a bunch of different things. These pictures on this page, following page, are all from panel 4-1 and give you some ideas of the kinds of things that proteins do. Now, as we know, a protein has the particular function it does according to the unique characteristics of its shape. The shape is very complex, and fundamentally that complex shape of a protein is specified by the sequence of amino acids. If we look at this panel, this is figure 4-3 from your textbook, we can see the names of the 20 common amino acids and see that some of them have R groups, also called side chains, that are negatively charged, whereas others have positively charged side chains. Some are polar and others are non-polar or hydrophobic. These properties in general specify much of the folding that happens as a polypeptide chain becomes a functional. In order to link two amino acids together, we need to look at not just the unique R groups, but also their common groups, common to all amino acids. Notice that both glycine and alanine at the top of the page have an amino group, positively charged, and a carboxyl group, negatively charged. A condensation reaction joins the carboxyl of one amino acid with the amino of the second amino acid. That condensation reaction, of course, releases a water molecule and it forms a peptide bond that links the two amino acids together into a dipeptide. Take a look at this short 4 amino acid peptide in this illustration. Notice that the side chains of the 4 different amino acids each have their own unique structures highlighted in the different colors. The backbone is shown in gray. If we look at the backbone, what we can see is that it consists of a repeating sequence of NCC, 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 and so on. The N terminus is how we identify the beginning of the peptide with an amino group or a nitrogen containing group. We always specify the end of the peptide chain as the C terminus with its carboxyl group. This orientation corresponds to what we know about DNA when we talk about the orientation being 5' to 3'. In proteins, it's N to C. Now pay special attention to the oxygen and the nitrogen atoms that are a part of the backbone because they're going to be very important to us as the protein folds. Interactions between R groups can help a polypeptide to fold in its own unique special way. Notice that these interactions may be electrostatic interactions, highlighted in yellow. They may be hydrogen bonds, shown with the pink parallel lines, van der Waals are very weak interactions, especially as we see here between these hydrophobic R groups. Hydrophobic R groups are especially important in polypeptide organization because they need to bury themselves away from the aqueous surroundings as they are repelled by the water. So the hydrophobic region tends to form the inner core of a protein, and polar or charged R groups surround them and form that direct interaction with the aqueous space around it inside. The stability of a protein shape is by and large produced by hydrogen bonding, which is very, very prevalent inside any protein's folding. That's because the backbone nitrogen and oxygen atoms, as pointed out previously, can participate in hydrogen bonds either with one another as a part of the backbone to backbone bonding seen here. They can participate in hydrogen bonds with side chains that are polar, as illustrated in yellow. Also, polar side chains are able to form hydrogen bonds between one another, so they all interact in stabilizing the. So this is a summary slide. You may want to pause your video on this slide and make sure you have all of these listed in your notes. The exact folding pattern of a protein is usually called by the word conformation. The conformation of a protein would be the folding pattern that has the lowest free energy, the lowest delta G for the conditions in which the protein is found. If those intracellular conditions change, the conformation also may change. Sometimes a protein misfolds. This misfolding is rare, but it can be quite dangerous. Misfolded proteins are called prions. An abnormally folded protein has regions exposed on its surface that actually should not be exposed, and that can cause them to aggregate or bind with proteins that were already folded normally, inducing a misfold in those proteins. Aggregated prions can form what we call amyloid fibrils or plaques. Plaques build up in tissues over time, and they can cause serious damage. For example, the plaques that are a distinguishing characteristic of brain tissue in someone who has Alzheimer's disease or another one of the degenerative brain diseases, as well as diseases that are found in other parts of the body. While some proteins are able to fold spontaneously, many of them incorporate the help of a certain class of proteins called chaperone proteins. The job of chaperone proteins is to inhibit misfolding. In this example, we're looking at one type of chaperone protein that will bind to regions of the polypeptide chain that may be especially prone to causing misfolding. The chaperones will let go of those regions after the rest of the protein has also folded correctly, so they resume their final conformation as the last part of the folding process. Another type of chaperone protein is illustrated in this picture. In this case, there is a chamber formed by a chaperone protein in which a polypeptide chain would be sequestered. Another part of the chaperone protein forms a cap or a lid. Inside this chamber, the isolated polypeptide is able to fold correctly without influences from other components of the cell that could cause it to misfold. Once the protein is folded properly, it'll be released and then can resume its function.